Hello all. In this video we are going to see about another phytohormone, that is Jasmonic acid. It belongs to non-classical class of phytohormone. Jasmonic acid is an organic compound. It belongs to Jasmonate class. Jasmonate class. It is volatile and it is present in essential oil, present in flowers and reproductive tissue, low level in root and in mature leaves. These are the basic facts about Jasmonic acid. It belongs to Jasmonate class. It induces senescence in plants, used in cell culture, causes degradation of chloroplast and loss of chlorophyll. It is induced during biotic and abiotic stress. It has anti-pest defense property. It stimulates ethylene synthesis. It stimulates fruit ripening. New facts. One phytohormone that is involved in the stimulation of another phytohormone, that is ethylene synthesis, is Jasmonic acid. What are the inhibitions that Jasmonic acid is associated with? Associated with inhibition of pollen germination, tendril coiling. These are and it is also involved in growth inhibition. Growth inhibition. Is involved in growth inhibition. Receptor. Now we are going to see about the name of the receptor. Is COI1, that is coronotic insensitive 1. So far we have seen about the basic details. What is the receptor involved, what are the inhibitions associated with, functions and basic facts about Jasmonic acid. Now we are going to see about biosynthesis of Jasmonic acid. Biosynthesis of Jasmonic acid. Precursor of Jasmonic acid is alpha linolenic acid. It is converted into peroxide. Peroxide is converted to allyl oxide. Allyl oxide is converted to 12-oxo-phytodinoic acid. Enzyme responsible for conversion is lipoxygenase. Conversion of alpha linolenic acid to peroxide. Peroxide is converted to allyl oxide with the help of enzyme allyl oxide. Cyclization takes place. Here allyl oxide cyclase. 12-oxo-phytodinoic acid. All these green letters occur in plastid. Plastids. 12-phytodinoic acid from plastid move into peroxisome. Where it form Jasmonic acid. by the action of enzyme beta oxidation occurs. This is a previous year question. An action of reductase. So, alpha linolenic acid is the precursor. It is converted to peroxide. Peroxide is converted to allyl oxide. Allyl oxide is converted to 12-oxo phytodienoic acid. Here the enzyme involved is lipoxygenase. Here the enzyme involved is allyl oxide. Cyclization occurs. Here allyl oxide cyclase. Rearrangement occurs. All these occur in plastid. Now 12-phytodienoic acid is converted into jasmonic acid. It occurs in peroxisome. Beta oxidation occurs. Reductase are involved. So, this is a previous year. This is a previous year question. Beta oxidation. This is a previous year question. Organelles name. Plastids previous year question. Peroxisome previous year question. Allyl oxide. Cyclization previous year question. Allyl oxide is converted to 12-oxy-phytodienoic acid. This is a previous year question. So, please make note of all the previous year questions. Next, we are going to see about signaling pathway. Signaling pathway will occur in two ways, that is in the presence and absence. We see when jasmonic acid is present, what will happen? When jasmonic acid is present, GAS, jasmonic acid, will bind with MEC2. Will bind with JA when jasmonic acid is absent. Case 1. Absence of jasmonic acid. So we are going to see what will happen if jasmonic acid is absent. We have signaling components like JAZ, MIXI and GA response gene. JAZ will come and bind to MIXI. MIXI will bind to JA responsive genes. So, no work, no function occur. So, no function occur. This will happen in the absence of jasmonic acid. So, in the absence of jasmonic acid, no function. In the presence of jasmonic acid, mix C2 GA response gene. So we have MIC2 and GA response genes. When jasmonic acid is present, GA Z will degrade. So GA Z will degrade and many functions will occur. Transcription, translation, whatever function occur. This is the signaling system of jasmonic acid. Let's sum up in our usual way, in the absence and in the presence. We have all three things: GAZ, MIC-C, GA response genes. We have MIC-C, GA response genes, and what will happen we will see now. GAZ bind to MIC-C2, then attach to GA response genes. No function. Presence of jasmonic acid, MIC-C2, GA response genes, GA degrade. Work occurs, function occurs. These are the basic differences. In the presence of jasmonic acid, function will occur. In the absence of jasmonic acid, function will not occur. These are the signaling pathway. Hope it is clear. With regard to jasmonic acid, we have seen about the basics, role and functions. We have seen signaling pathway, and we have seen biosynthesis of jasmonic acid. Hope this video is clear and this video is very useful. Thank you for watching this video.